Hello, nice to see you. Today we're going to do a question on non-coding DNA. The reason why this video is just going to be on this one question is because if you truly understand how or understand what non-coding DNA is, it really trickles into other questions from this topic of manipulating genomes. So this is a standalone because I want us to really focus in on this. If you can understand this fully, you're on the right track. So pause the video here, try this question out, and continue on to see how I answer it. Okay, so explain why only selected sections of non-coding DNA are used when profiling a human. Okay, number one, what the hell is non-coding DNA? It's the part of the DNA that does not code or make a protein. And if you would like to know, approximately, I'd say 3% of the whole genome is non-coding. Okay? And essentially, the way I would rephrase this question is like, okay, why are we using non-coding, but why aren't we using coding sequences to profile a human? Okay, each human's different, but actually, funnily enough, the genome is very similar between me and you and your cousin, you and your mum. Like, we're all very similar. I mean, look at us. We've got the same, like, I'm saying this with asterisks, right? But like, we have hands, we have legs, we have feet. We like, you know, we all basically look kind of like the same, but there's variations, right? Because of sexual reproduction and stuff. But the genome is very similar, and so if you're using coding sequences, well, you can't really profile a human, like, let's say person A, because there you wouldn't get any unique profiles. What that means is like unique data or like repeating sequences of that individual. At the end of the day, right, DNA is based on codes, based on bases, and so if you don't have, specific repeating sequences, then you can't really identify between person A and person B, right? And the thing is non-coding DNA, it contains short-handed repeats, aka repeating sequences. So I think it's to do with Parkinson's or Alzheimer's. It's in the textbook really, but they've got a specific number of repeating sequences, and that once you see it, not like visibly see it, but once you see it, you can identify whether that person has a disease or not. And so that's like one of the really useful ways that we're using repeating sequences in non-coding DNA. So when you get a question on non-coding DNA, maybe, I'm not going to say it works 100% all the time, because there isn't many questions that I've found personally on non-coding DNA at this moment of time really, but try and rephrase it in a way that like is the opposite. So here I rephrase it like, okay, why aren't we using coding DNA? Why are we using this one instead, like non-coding? And then hopefully, you can get to the answer nicely. But remember, each cell contains the whole genome. Okay, every cell in your body contains the whole genome. Every human genome is extremely similar. 97% of us is similar, 3% is different. This is approximations here. So yeah, I would recommend you write this down in your notes and then really try and internalize it, Search on YouTube about, okay, non-coding DNA. Just look at these DNA sequencing people, like the actual companies have videos, like animations on YouTube. They're so useful to like understand, but also really be able to picture it in your head. So that's something I'd recommend you do after this video. And yeah, remember, the two most important exam techniques are read the question first and state all the obvious points. Now over to you. Try some questions and let me know how it goes.